Hi, this is Miss Litton, and these are my wonderful, these are my wonderful APBio students. Say hi. Hi. They're super excited, and it's lunch. Okay, so I want to go over like some main things here, but I also want to deal with the essay questions relating to this particular chapter. So if you haven't started writing them, you could. So the first things first is you would have to be able to identify the parts of a membrane and the role that each part plays. The parts of a membrane and the role that each part plays. So if you have started some group shared notes for your review right now, see what you can come up with as teams before I go over it. Okay? Name each part and any role that it plays. And I'm going to pause just for a second. Okay, so I'm going to interrupt wherever you are in that process, and let's start going through it. So the bulk of the cell membrane is made out of two different items. What are those two different items? Perfect. Phospholipid bilayer and proteins. So the phospholipid bilayer, it's creating a barrier, so what's happening to the interior of the cell is different than what's happening to the exterior of the cell. Why can it do that? It's a phospholipid bilayer. So how many layers? Two. And then let's analyze what those phospholipids are like. Okay, they have, we know lipids in general could be what's and what's if you're a lipid. Fats or steroids. So let's do the fat structure. A typical fat is built of what? Attached to glycerol. So you have 3 carbons, and then you have these 3 fatty acid chains. In a phospholipid, what have we done to one of those chains? We've removed it and instead replaced it with a phosphate group. That's phosphorus with some oxygens around it. That part of it, okay, is the phosphate group is hydrophilic, whereas the fatty acid chains, those are like hydrocarbons, right? That is hydrophobic. So when you have the two chains, then the middle part here is that hydrophobic region, and then the phosphates are in the hydrophilic region, okay? And what's going to prevent something from crossing the cell membrane is this region right here, the hydrophobic part, okay? So it creates a barrier. What was the second thing you told me you would see in a cell membrane? Proteins. So proteins play various roles. Let's review them together. Ready? It could be a what? Channel. Okay. It could be a carrier. It could be a receptor. Right? I don't know what order I gave them to you. It could be what? Enzymatic. It could be cell recognition. And what else? Did we hit them all? Yeah, well, you can have junctions. Use them for junctions. Yes. Okay, so it plays several different roles. And we'll go into each of those roles in a little bit more detail. Now, there are more things in there. Okay, so we took care of our phospholipids. We took care of our proteins. What's another item I would see in a cell membrane? Cholesterol. Good. And you can see cholesterol right here. Okay, and cholesterol is for what? Stability. Right. Now, is cholesterol found in all cells? No. Animal cells, right? Animal cells. And the cholesterol is there when it's too hot, kind of keeps it all together, and when it's too cold, it prevents it from freezing. Good. Fourth item. What's the fourth item you see in a cell membrane? What? Extracellular matrix? Is that what you're telling me? Good. And that's part of your glycocalyx. And there are 2, glycolipids and glycoproteins. And that's just carbohydrate chains either attached to a protein or a lipid. And that helps with what? Cell recognition. Tells you if you're too crowded, if you should be here or if you shouldn't be here. Okay, helps with development. Okay, so knowing the 4 parts, being able to recognize them and the role they play, that's critical, okay? It's called a fluid mosaic model because the proteins are migrating around. Remember that it has like olive oil. It's about the consistency of a cell membrane. Okay. A cell membrane doesn't look the same to the interior as it does to the exterior, because the exterior, what do you have on your cell membrane? Your carbohydrate side chains, your glycocalyx. To the inside, you have part of your cytoskeleton. Good. Okay. Tell the person sitting next to you, on a scale of 1 to 10, how well do you know and understand the 4 main components in each of their jobs? Go ahead. All right, and then I'm blazing through this really quick. We just said channels, like moving chloride ion from one side to another. Are proteins specific? Yes, very specific, always. So we have channels, and then we had carriers. Okay. We talked about cell recognition with your glycocalyx. We talked about receptor proteins. And we talked about how they can catalyze enzymatic reactions, and we also talked about junctions between. Okay, now we know the structure. I want to get across this membrane. There are 3 ways to get across. Don't tell me out loud. See if you can type it up right now. 3 ways to get across that membrane. 3 ways to get across. And why you go with each way. See if you can figure that out. The 3 ways and why you would go with each way. Okay, just so we can get through everything, I'm going to keep going. Okay, one way is what? Through the phospholipid bilayer, but you cannot do that if you are larger charged, because that hydrophobic interior is going to repel you, right? You can't do large because you can't fit in between. Okay, so through the phospholipid bilayer, or using a channel or a carrier. Okay, that's way 2. And way 3 is, yeah, whole membrane. Through the phospholipid bilayer, a channel or a carrier, and using the cell membrane. Now, we can take all of the transport and we can categorize it into 2 big categories. What are the 2 categories? Those were the ways to get across, but we can categorize them by if they what? Active or passive, if they cost us energy. So see if you can come up in your little group shared notes. What are some examples of the passive transport, and what are some examples of the active transport that we discussed? See if you can come up with it on your own. All right, so I'm jumping in. Passive transport does not require any energy. If you're doing this, diffusion right through the phospholipid bilayer, that's not going to cost you any energy. That's free of charge. Okay, if you use a what? Channel, and you're going from a higher concentration to a lower concentration, what is that called? Facilitated diffusion. Is that going to cost you anything? No. Okay? When you're just using a protein as a channel because you are either larger charge, but you're going with your concentration gradient, that's not going to cost you anything. Oftentimes, carrier proteins are pumps, but there are some carrier proteins that are not going to cost you as long as you're going with your gradient. If you go against the gradient, that's going to cost you energy. You're concentrating to one side of the membrane. That is called active transport. There's another kind of active transport as well, using the whole what? Membrane. Using the whole membrane. Okay, and then we looked at some examples. We looked at an aquaporin, which is a type of channel protein for moving what? Water, so it can move readily. So here, 21 number. Okay, I'm done. Okay, close. Okay, carrier proteins are always specific. Okay, all right. Cholesterol is found in the what? Plasma membrane of animal cells. Sorry, I just wanted to go fast on that one. Okay, then we talked a little bit about introducing signal transduction. That's going to come up when we talk about hormones, but I wanted you to see where a protein is acting as a receptor, and that's setting off several relay molecules, which then you have some sort of response in relationship to that. And we'll spend some more time talking about that later, but next I want to talk about diffusion, and most specifically osmosis. Diffusion is just going from a higher concentration to a lower concentration, but osmosis is the diffusion of water across a semi-permeable membrane. All right? So let's talk about a couple different scenarios here. So you have a beaker. Here are 3 beakers. And we're going to put water in each of them. Okay? And then we're going to put some salts and some salts and nothing. And we're going to have red blood cells. Oh, I need some space. Yeah, I don't want to hear about it. Okay, we've got some red blood cells there. Now, I have given you 3 different scenarios here. I want you to talk about things like hypertonic, Okay. Okay. I want you to talk about things like hypotonic, hypertonic, isotonic. Describe what is going to happen to the red blood cell in each of those situations. Go. All right, so that's all I have patience for. What kind of solution is that? Hypotonic. In relationship, the red blood cell would be what? Hyper, and you know water must flow from the hypo. What will happen to that red blood cell? Burst. Yeah. Now, would the same thing happen if it were a plant cell? No, because it has a cell wall who would push back, and that's called what? Turgor pressure. And then I can't help but think about this little equation, right? Okay, so that's a factor in the way water moves, is the pressure potential, the cell wall moving back. Okay, in this situation, what is the solution? Hyper, and in this case, the red blood cell would be hypo. Water must flow from the hypo, so water would flow out, and what would happen to this red blood cell? Shrivel up. Would that also happen to a plant cell? Yeah, you would still have the cell wall, but what would get smaller? The vacuole, and then the whole cell would kind of shrivel down, okay? What about in C? What do you think C is? It's not called nothing. Isotonic. Same. Okay? Relative to each other. So does that mean water is not moving? What does that mean? No net change. The same amount moving in is moving out. Perfect. Okay. All right. Okay, and then we look here. Okay, and we've already addressed this as well in the lab, and we talked about why substances would move, and they're a factor of two things: pressure potential and solute potential. So this cell is not going to get any larger because the osmotically active substances that would bring that water in, you have the turgor pressure generated by the cell wall that's pushing back out at the same amount, so there's not going to be any net change. Okay, close. Water is always going to be the solvent, and whatever you dissolve in is going to be the solute. So when you say hypertonic, you're really saying hypersolute, right? Okay. It will. Yay! Okay, now, we have already talked about this. We've already reviewed it. You can use a protein, and it's called facilitated if whatever you're moving using a protein is going what? With the gradient. Good. If you're going against the gradient, it's called active transport, and our best example of that is the sodium potassium pump, which pumps 3 sodiums out for every 2 potassiums in. Yes?
What exactly is the gradient? Is it a structure or is it the way the...
Oh, okay. So when we talk about the gradient, okay, see these orange balls? Where do you have more orange balls? Outside. Okay? So there's a higher, when you think of like a grade, like on a road, there's a higher amount of orange balls on this side than this side. So they'll want to go in this direction.
That's considered the gradient.
Yeah, that's the gradient. It's the solute that you're looking at. The concentration of the solute is the gradient.
Okay.
Could the sodium potassium pump be going with the gradient like at instance?
Okay, not the sodium potassium pump. The sodium potassium pump is built to pump sodium out, 3 out, and 2 potassium in. Now there are sodium gates and there are potassium gates that allow it to go the other direction, but they're triggered differently. And we'll be talking about that. Okay, then last but not least, we talked about using whole membrane, endocytosis and exocytosis. And we said that there were 2 types of, actually 3 types of endocytosis. See if you can come up with those on your group shared notes and how you differentiate between them. Okay, what are they?
Penocytosis.
Penocytosis is what? Cell drinking. What's the other one? And what is that? Eating. And one more. Receptor-mediated endocytosis. And remember on receptor-mediated, it goes ding, 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 ding on a bunch of receptors, and then it engulfs. And the opposite of endocytosis is exocytosis. Okay, and we're just almost done, and then I want to go over the essays. And you should be good on those essays based on our review. Okay, let's go. Okay, is pinocytosis, somebody picked B, is it a form of endocytosis? Yes, but so is everything else. Go, peeps. No, you're not. Ah, maybe you are. Okay. So somebody said cholesterol. Pumps are not made out of cholesterol, but cholesterol are in animal cell membranes. My hand at the shower thing. I can see me like. Ah! Pickles appear wrinkled and shriveled. Now, pickles, what were pickles before they were pickles? So did they get bigger or smaller? So water must have gone out of them, right? Water must flow from the hypo. So if they got smaller, water came out of them, so they must have been in a hypertonic environment. Check with your bio buddy, see if they got that right. All right. Cell junctions, we talked about the different, here we can just use this one right here. Okay, tight junction, be a tight junction, that's protein to protein, right? To stitch it, it's like stitch between the cell membranes. What's this one? What's this one? Gap junction is channel to channel to channel, channel to channel, and then what? Adhesion. Cytoskeleton to cytoskeleton. Okay, we talked about plant cells. They have their cell membranes. You have to have an opening, a doorway in the cell wall, much like a gap junction. Okay, and that's called plasmodesmata. Do as you oughta. We already talked about that. Okay, I want to talk about your essays real quick. Okay, potential essays. Okay, number 3. Okay, number 3 says, describe the model of the cell membrane of a eukaryotic cell. So the model of the cell membrane, what is that model called? The fluid mosaic model. Fluid mosaic model, good. And then it says, and discuss different ways which substances move across the membrane. In your discussion, be sure to indicate whether your examples are passive or what? Active. So you would describe the structure of it, the phospholipid bilayer, the proteins, the cholesterol, the carbohydrate side chains, and then you would talk about the 2 different categories and examples of passive and active transport. What are the examples of passive transport? Okay, osmosis, diffusion between the phospholipids. Yes. What's another example? Facilitated transport using a channel. What are your examples of active transport? Pumps and whole membrane. Yeah. And you would describe the difference, why it's passive, why it's active. Okay, this number 4, this is basically your pre-lab for your lab, right? Look at this. A laboratory assistant prepared solutions of 0.7, 0.5, 0.3, and 0.1 molar sucrose, but forgot to label them. After realizing the error, he took off to Mexico, and then his assistant randomly labeled the flasks of the unknowns A, B, C, and D. Design an experiment based on the principles of diffusion and osmosis. If something says that, based on the principles of diffusion and osmosis, do you think you should first start by defining diffusion and osmosis? Yes. Okay. That the assistant could use to determine which of the flasks contain each of the 4 unknown solutions. Include in your answer. So let's just take a couple minutes to talk about this. Was your lab. Remember you took your dialysis tubing and how you put different molarities of solutions in it? Right? And then we looked at the results and we looked at the trend. What was the trend that we found? The higher the molarity, the greater the percent change in mass. So if you took these solutions, unknowns, put them in distilled or put them in dialysis tubing and then put that in water, weighed them before and after, you would expect whichever one gained the most mass had what? The highest molarity. Whichever one gained the least would be the, yeah, so based on lining them up, most mass to least, you could say highest molarity to less, right? Sugar goes in the dialysis tubing. Okay? Make sure to start writing these essays, guys, because that test is going to sneak up, right? Okay, you're super smart. I'll see you mañana. Do your highly suggested reading and thinking. Remember to keep up with that as well. Be smart.